Dr. Laura Hernandez is an associate professor in the dairy science department at the University of Wisconsin-Madison, so she's one of ours. She's also a member of the Endocrine and Reproductive Physiology Interdisciplinary Graduate Program in Nutritional Sciences and the Comparative Biosciences Graduate Programs. So basically, she does a lot of translational stuff working with our reproductive physiology and endocrine group. So the people that study infertility are working with her in her department, which is great. She received her PhD in 2008 from the University of Arizona and completed her postdoctoral in molecular and cellular physiology at the University of Cincinnati College of Medicine. Her area of research has focused on how serotonin controls the mammary gland and various aspects of lactation. Dr. Hernandez combines basic research from the cell to whole animal level in a variety of mammalian species to broaden the focus on the importance of the mammary gland and its contributions to and regulation of a successful lactation in dairy cattle, and also for the brains of all of us in the world. I just had to add that. The outcomes of her novel research are aimed at demonstrating the presence of factors, specifically serotonin, produced within the mammary gland that can control the animal's physiology while lactating, particularly during the transition period when cows are at the most metabolically and physiologically challenged. She specifically focuses on the interaction of serotonin and calcium metabolism during the transition period. Her research has determined that serotonin is an important regulator of mammary gland, maternal calcium, and maternal energy homeostasis during lactation. She has authored or co-authored 33 peer-reviewed journal articles. Her research on the coordination of maternal metabolism during lactation by the mammary gland has numerous applications to women who are breastfeeding and is focused on improving maternal health during this time frame and in later life. So I'm pleased to, it's my pleasure to introduce Laura Hernandez.
Thank you for that introduction, Dr. English. Just so you guys know, I'm a little bit newly sleep deprived. We got matched for adoption with our second baby, and I know there was a couple of the NICU nurses that found me at Meriter a couple of weeks ago and were like, "You're speaking in a couple of weeks, are you still gonna do that?" And yes, because I'm really, really excited about this. We do work with dairy cows in my lab, but we also work with rodents and are really trying to get our work translatable to human medicine. And so I was really, really fortunate to meet Dr. English very early in my time here, and we've slowly and surely been getting closer and closer to that. Today she's asked me to talk about two kind of broader areas of lactation biology, and actually my grad student Sam Weaver will be talking about what we've been doing with SSRIs and serotonin and calcium in her talk, because it's been a very large part of her research. So I'll leave that part to her. So today one of the things Dr. English wanted me to talk about was feedback inhibitor of lactation. Sam and I wrote a review paper that came out in 2016 in the Journal of Dairy Science discussing how the mammary gland in the baby and the milking machine in the dairy instance, or our milking machine in the rodent instance, or the pups in the rodent instance, are driving milk secretion and synthesis. And for a long time there was this notion of this chemical called feedback inhibitor of lactation, which as I didn't really know because I wasn't as familiar with the human medicine literature up until the last several years, is still kind of a big concept. And it is a big concept, but FIL itself, as you'll see when we go through these slides, isn't the actual thing. And actually FIL itself as a chemical kind of got debunked about, I want to say 20-ish years ago, and my PhD advisor was one of the people that went and was like, where is this protein? What's the sequence? What is it actually? And what I'll show you is that it's probably a lot of different proteins working together to drive milk synthesis through this input through the baby. So how is milk production homeostatically controlled? So the mammary gland is really cool because when an animal is born, they're all the same, just about, really. And it's the only tissue in the body that really changes postnatally. And that's what makes it so amazing. And we can actually manipulate it in a lot of different ways as a result of this that allow us to try to get at what's happening in the mammary gland because it develops postnatally for the most part. So what you see here is a mature virgin tissue, and this is a rodent. I'm sorry, cows are also difficult to get tissues from, and clearly humans are very difficult to get tissues from. So a lot of this research is in rodents because we can get lots of tissues from them. So you have a mature virgin tissue here, and you can see these are some ducts, and these would be terminal end buds where you would have a lot of the outgrowth of the mature tissue once an animal becomes pregnant. You do see some tissue growth during puberty. There will be a little bit of growth that occurs in a human, in a rodent, in a cow, but the majority of it is going to occur during the pregnancy phase. And you can see these terminal end buds here become very large. They start to engorge, they start to proliferate and grow, and they look like kind of these big clusters of grapes. And this tissue organization actually, surprisingly, is quite similar to a lung. It looks very similar, and the way the fluid moves is there's a lot of similarities. Once you get into a lactating tissue, you can see there's tons and tons and tons of tissue taking up this area in the breast or in the mammary gland or in the udder, depending on what species you're working with. And then typically what's going to happen is that the tissue is going to regress to look back kind of what it looked like in this mature virgin state. So it'll happen as long as you're not a concurrently pregnant animal, which would be a dairy cow or a dairy goat, or maybe similar to this woman that's had 20 children and has been pregnant for most of her life and has probably breastfed, but it's very much not a normal state. So usually what we'll see is the tissue will go back to normal. And what we're going to talk about hopefully in this lecture is to kind of get through how do we maintain this? Why can we maintain it for so long? Why can a woman breastfeed for 5 years without having this pregnant state to feed back with these hormones? And hopefully what you'll understand is that you do have to have the endocrine hormones to set up the tissue, but it's really the baby or the milking that's driving this continued secretion and synthesis of milk by the breast, and it's really this kind of locally controlled situation. So typically we think about developing tissue in the breast and during lactation in this classic kind of endocrine format. And here we have the hypothalamus, and the hypothalamus is going to regulate the pituitary gland to make a lot of different hormones that are going to work together that all contribute to lactation. So it was mentioned earlier when you're thinking about mom and there's a stress and potentially you can see low supply. Well, it's because the stress hormones are also involved in making milk. So there's a lot of things going on that need to keep supply up and keep mom happy and keep lactation going forward. So what we have here is prolactin and growth hormone IGF-1, more so in the cow, are the primary kind of galactopoietic hormones that are going to keep lactation going forward. So often in humans, we target prolactin if it becomes that dire situation where they're just not making enough and they want to keep up. I fell to that with my first child. I was for sure going to make it a full year because I'm a lactation biologist and I was a new assistant professor, and I was like, I'm gonna do this. This was the first year I was teaching lactation physiology, and I was a bigger mess than I am now. And I made it about 4 or 5 months okay. But given all of those things, I started to kind of fall apart, went in to see my clinician, and I went on to the medication because I was going to make it a year. I made it 7 months, and then my husband was like, you're done, you're falling apart, and this isn't good for anyone, particularly probably him. But I made it as far as I could, and I was really bummed because I really wanted to do it, but that's where a lot of our research comes from. What about mom? And we're really focused on trying to understand her biology better to support her to lactate. So as you can see, this is ACTH, which is going to stimulate the adrenal glands, and cortisol is a very important component of lactogenesis. Thyroid hormone, which is affected also by the anterior pituitary, really important for lactation as well because it's going to provide the energy in using ATP, and lactation is a very energy consuming process. And clearly the ovarian hormones are really important during the pregnancy to set up the tissue to have adequate ductal branching and proliferation of those cells and proliferation of the epithelial cells to make sure we have enough to stimulate the lactation once the pregnancy comes to an end. So the endocrine portion is super, super important during the pregnancy to set up this tissue. And it does still have some importance during the lactation, but really the mammary gland takes over after that. So all of these hormones are working together to make sure we're making enough epithelial cells because milk production is a function of mammary epithelial cells. So you do have to have a good pregnancy with the right setup of hormones and the right endocrine status to have adequate numbers of epithelial cells so that when you do start lactating, you have everything that you need to kind of have a successful lactation. So this is just as important as the lactating part of the story. And then clearly the posterior pituitary is also going to be really important with oxytocin and coordinating milk letdown. And that's really important also in the maternal fetal bonding. You hear a baby cry, your milk lets down. You're talking about milk letdown in your lactation class. As the first year you're here, you will let your milk down. It's very bizarre, but it happens. And that's not just the letdown part, it really is part of the bonding with the baby that facilitates this as well. So all of these hormones are really important. And I always tell my students, you ever wonder why a woman after she gives birth might be kind of crazy? I was one of them. Think about all of this. So it's a lot going on at the same time, and it needs to be set up appropriately during the pregnancy and during the bonding stages so that you can have good letdown and good milk production. So that's the pregnancy part. What happens when you actually start lactating? Well, as I said, the mammary gland really starts to take over during the lactation phase, and we call this autocrine paracrine regulation, and that's because the cells in the tissue make things, hormones, growth factors, that are going to get into the milk. And if you don't milk often enough or you aren't expressing milk a lot, the best way to start to increase your supply is to either pump or feed more or milk more in the dairy situation, because you're removing those chemicals, you're removing the physical component, which is the stretch or the increase in volume within the tissue, and those are going to send signals back to the tissue that says we're empty, let's make more. So we have this endocrine stimulation that's critical to set up the tissue structure, but really it's this local control within the tissue that takes over during the lactation, and those can be both positive inputs or negative inputs depending on what you're dealing with. And that's where the focus of our research is. So what is local control? Well, there's factors in the mammary gland that regulate milk production. The big one for a long time that was talked about was FIL, or feedback inhibitor of lactation, and it was really based on if you didn't milk an animal only once versus twice daily, the supply would go down. There's a stretch factor within the tissue. You can only accommodate so much fluid in that space. But there's also chemicals that are going to be building up in the milk that can send signals back saying, let's slow this down because we're not removing the milk. So the amount of milk removed and the frequency are critical to milk yield. And this is not a species-dependent phenomenon. This happens in rabbits, it happens in wallabies, it happens in cows, it happens in humans. It's a part of being a mammal. And the systemic hormones are really largely uninvolved. I mean, you have to have, in most species, enough prolactin around to kind of keep things going. And in cows or other ruminants, growth hormone has to be at a certain level. But really beyond that, you could take that all out and just kind of keep going, and the mammary gland is going to take everything over. And in fact, it will dictate to the rest of the body, I don't care if you didn't eat enough, I'm still going to take that fat off of your back, or I'm going to take the energy from your liver, because really what we need is to make this milk, and that's more important. So I always tell the woman who works on the liver in our department, you might need the liver, but really the mammary gland's in control when we're lactating, so that's just not that important. And we feud about this. So, but really, it's this tissue that's dictating what needs to happen. And so the mother is sacrificing at all times for this. And so really where we focus is trying to how do we can improve that and make it easier, because it is a demanding process. So my favorite mammal are the marsupials, and they are the most exquisite example of local control. So these guys, whether you're talking about a wallaby or a kangaroo, they have a really interesting reproductive life cycle. So they have their babies at about 21 to 28 days in gestation. And we were talking about earlier about the baby crawl. These animals, when they're born, literally crawl out of the vaginal canal into the pouch and attach to a teat. And so that's like probably the primitive baby crawl, because they evolved before we did. But what happens is you can have an animal or a newborn wallaby in this case, and they're the most studied laboratory marsupial, is born, and you can see they're very, very, very small and immature, and they're very underdeveloped. And the mother is going to be making a certain amount of milk that is very much critical to this newborn baby. At the same time, on a different gland, she'll have a joey that's over here, 150, 200 days old, and is out foraging, coming back, and the milk is completely different in composition. And this is the finest example of what the mammary gland can do. It's tuning to the baby and the baby's needs at the same time, irrespective of what the mother's eating or how she's living. She's got one milk over here for this baby and milk over here for this baby at the same time. Completely different composition. And that's the mammary tissue adapting to what needs to happen for the baby. And I will beg and beg and beg my boss to bring these over here, and I'm sure he'll keep telling me no. But they're really just fascinating animals. And between platypus and the egg layers and us, this is the link. They're kind of in between the mammals that are the platypus that still lay eggs and secrete a fluid along their belly without nipples, and a human or a cow or a rodent or whatever. So they really, really have this very different milk composition. So you can see here the really high carbohydrate at this early stage, lower protein, lower fat, and then you get over here and it flips, and you have higher lipid, less carbohydrate, and moderate protein, because this animal isn't nursing as often, so they need more of a dense caloric milk because they're foraging. And this one is constantly nursing. So their lipid's gonna be a little lower, carbohydrates higher, they need energy to develop into this joey. And same time, every time I think about it, it really just kind of blows me away. So a lot of the work has focused on this, on how the local control of the mammary gland has really kind of gone forward. So I kind of have this timeline here of how this all falls into place. So in 1984, this has been observed in lots and lots of different species, but they did a study where they saw that milking frequency didn't change milk composition or mammary blood flow. And this was done in a goat. So everyone was trying to figure out, if we nurse more often, are we changing the composition? Is it blood flow? Is it a hormone? Is it the tissue? They really didn't know. So this suggested that whether you milk them once, twice, or 3 times, you were still having lots of blood flow to the tissue, and the milk composition was the same. So that was a clue that something in the mammary gland was allowing them to produce more milk. And again, this was in the goat. Then in 1986, this is one of my favorite kind of studies. This was done in humans, and they had a cohort in Gambia and a cohort in the UK. And these women were making the same amount of milk. The women in Gambia had far less access to food, different socioeconomic status. The women in the UK had whatever they needed, kind of your run-of-the-mill, middle, upper class type family. They were milking twice a day or nursing twice a day, and the women in Gambia were nursing 15 times a day, making the same amount of milk for the baby, and the babies were still growing. So again, another example of it's not really what you're eating. Clearly, you'll do better if you're eating more or you have more access, but if you're undernourished, you can still feed your baby as much as they need to feed, and the mammary gland will extract those nutrients from somewhere. In 1959, that was the first evidence that milking frequency in like a lab situation became really apparent. They looked at some various routines of milking frequency and basically showed, and this has been shown over and over again in many species in the dairy side, if you go from a 2x, so milking twice a day, to 3 times a day, you'll increase milk yield by 10% without fail. And it's just a matter of removing more and removing something, and at this point they didn't know what, that was driving this. In 1983, what's neat about dairy cows or dairy goats is that, and in general, mammary glands are separate. So a human has 2 breasts, those are 2 different mammary glands. If you get an infection in one breast and it just becomes too painful to deal with and you can't get over it, you could dry that one breast off and keep nursing on the other side with no problem. And we do that in the dairy industry all the time. There will be a cow that gets mastitis in 1 quarter. They dry it off and they keep milking her on the other 3. And so because of this, we can utilize these half udder models that allows us to take away the variation between animals. So in a cow, we can give a treatment on half of her udder one way and then a different treatment on the other side. And in this case, we do this with frequency studies. So you can milk half the udder twice a day and half 3 times a day. And you'll see you can apply these treatments and you remove the endocrine inputs because you're just looking at what the mammary gland's doing, because the endocrine status is the same and the nutritional status is the same. And so we've done a lot of studies looking at this to try to get at what are the actual factors being driven when we were trying to figure out what is fill. Is fill one thing? Is it 100 things? Is it 200 things? And what they saw was that you still saw an increase with 3x versus 2x on that half udder, and it really didn't matter what the intake was. It was driven by the mammary gland. In 1982, they didn't use a half udder model, but this was the first time in dairy cows they showed that this 3x, so increasing milk yield 3 times versus 2 times, would give you a 10% increase. And as lactation consultants and people that are out there working with moms, the first thing they said to me when they came into the delivery room when I said I was going to breastfeed was, you have to just keep nursing and your milk will come in, and you have to do it more frequently if you're low. And this, even if you're having a hard time, you can nurse and pump after to stimulate some more flow if you have low milk supply. And this is just classic, more often, more milk will be made. And it's because the mammary gland controls itself. In 1980, what they showed was that if you went these long intervals, they figured out how long you should go between basically milkings in a dairy situation. 12 hours is about, where you're okay. Otherwise, you get to about 16 hours and you'll start to see decreases that can't be recovered. And similarly, I would assume in humans, there's probably a threshold of where you should not get to pump or get to nursing where you'd start to see some permanent decreases that potentially you might not be able to recover, particularly as you go further and further into the lactation. In 1996, they were looking at how to figure out what were the ways to stimulate this early on. Clearly, when we first come into milk, this is a little bit slower. It takes some time to get your milk yield established. The same is true in a dairy cow and same in a goat. And they were trying to figure out how they could get milk yield up quicker in that early time frame when you've just given birth, you have probably not eaten much at the end of your pregnancy. Same thing happens in a dairy cow. They give birth, they start lactating, and you're trying to get your feed intake up, but you've just given birth, you're stressed, you're tired, you're trying to nurse this baby, and so they're trying to optimize kind of milking strategies that would help the milk come in faster and go up quicker early on. And so they did a number of different things where they would do like 6 times milking for a couple of weeks and then switch to 2 or 4. And so there's different ways you can manipulate that first part of their lactation cycle. In a cow, it'd be 3 weeks. It might be shorter or longer in a human, but you can do more stimulation early on to help kind of bring them up. And then, as I said, with these different, because the tissues are separate from each other, the glands are separate. They've gone on to look at various different ways, 2x versus 4x, lots of different ways to increase the milk yield, but also in the cow where we've done biopsy studies to try to understand at the molecular level what's happening, what are the actual signals that are driving this increase in milk yield that will occur. And so the one thing also that has become very clear through all of this is that the persistency of the lactation, so the ability to carry it on for a long period of time, is really a balance of proliferating epithelial cells versus cells that are dying. So if you can keep stimulating the tissue and stimulating the signals that are going to increase the proliferation of those cells that are the fundamental cells to make the milk, and that remains higher than the cells that are dying, you can keep going. And that is really the take home of all the endocrine and as well as local stimulus factors, is that you really want to drive that. So what happens with milk accumulation? This is one of the things that is going to really drive suppression of milk, and it's going to be twofold, chemical and likely physical as well. Increasing milking frequency, when they showed that this increased milk yield, was largely due to this less milk accumulation. Because the more milk you have accumulating, the more chemical concentration of whatever is in the milk, which was probably several things, as well as the force or the capacity of the tissue to accommodate. There's a point at which it's physically like your gut, it fills up and it's too full, and it's going to put some pressure back that says, we're either going to explode or we need to slow this down because it's just too much. And this is where potentially, you can get into edema problems, and we talked about massaging the tissue. The same thing happens in cows. When we see like a first cow, we call it first calf heifer, she gives her birth for the very first time, she can swell quite dramatically. This would be seen probably for a primiparous mom. This is the first time she's coming into milk. The mammary gland has no idea what it's doing, and it swells, and it's full of fluid, and it's got more fluid it's making to make this milk, and udder massage happens all the time. So it was neat to kind of see that in application too, because we do it on farm all the time, and that's because the lymphatic system doesn't move without physical movement. It's not like you either have to breathe to make your muscles move, to move the lymph vessels, but they don't move without any kind of actual physical movement. So this milk accumulation that occurs, we do know it's independent. For a long time, they thought oxytocin had something to do with it. It wasn't your milk letting down. Oxytocin doesn't have anything to do with it. It's just that you're making more and more milk. And sometimes you can have a defect in milk letdown, but that's pretty rare, at least from a dairy perspective or in a rodent perspective. So they figured it had something to do either with the distension or pressure in the gland or some kind of chemical signal that was feeding back. And one of the big things is its effect on the tight junctions between the cells. So the mammary epithelial cells, like the gut, they have to get really, really tight to keep the milk in and keep that blood-milk barrier separate from each other. And this is why if you have a C-section or struggle with delivery or deliver the placenta late, you can have issues with this tight junction closure, and your milk takes longer to come in because part of it is still leaking out. And this is because two of the things that have to happen for them to close is the progesterone has to drop, and your cortisol level also has to go up enough to trigger lactogenesis stage 2 or secretory differentiation to occur or activation. So we'll see this in dairy cows who, if they have a retained placenta, they have a hard time as well because there's still a progesterone source kind of hanging around. And this milk accumulation within the gland, we do know is not the sole source of decreased secretion. So we know there's chemical factors that are occurring, but likely, again, this physical stretch that's a lot harder to look at in a lab, because you have to figure out how you're going to stretch a tiny little cell to try to get that feedback back. But they've done studies in goats where they used a bellows and they've injected air into the actual quarter of a goat, and they'll see changes in secretion as a result of that just because of the stretch. So one part is stretch and 2 part is chemical. So The first time they thought that there was a chemical present in the milk was in 1963, and for a while they thought it was a fatty acid that was doing this. And like I said, we'll talk about several different things that likely are. So how do cells turn over during a lactation? If you look at a lactation curve, this just happens to be a dairy lactation curve because we have a very standardized time in which they milk. But this is a lactation curve that could be applied in any kind of time frame for any kind of mammal. You're going to have a rapid increase for a period of time. And during this time, this is where I was talking about, you're trying to get them to come into their milk faster and make more milk. And there's a lot of struggles going on, from the birth to adequate nutrition to number of times you're milking. And then you hit a period where they're at their peak, and they're going to make their most milk that they will ever make. And then there will be some kind of persistent decline that occurs as the baby is getting older, or the wallaby's aging, or in this case, the dairy cow or the dairy goat, they're concurrently pregnant because they're going to have to come into a next lactation, which is something that's different than all other species, basically, because, or even beef cows, because they're not concurrently pregnant when they're lactating. So that's a little bit of a different situation. And then they're going to have this period of recovery in the case of the dairy animal, where they're turning over their cells and getting ready for the next lactation. But even in a human, if you're going to come on and lactate later again, you want to have adequate recovery of the tissue so that it's best able to adapt to the next pregnancy. So this dry off period, or the period when we stop nursing, it needs to happen in such a way that the tissue actually can repair and regenerate itself very well so that it's ready to go again. So as I said, this half-udder model is really useful because we can study how some of these things happen, and we can take samples from each quarter. We can look at the milk composition, we can take tissue biopsies and try to figure out what the science is behind driving this. And actually we're starting a study like in 2 weeks with a quarter-level milking device that this engineer built for us, and we're going to manipulate this and try to understand what happens if you leave so much milk behind, if you milk more frequently, and how this coordinates to really get at what's actually going on. And if there's ways we can improve that in any situation, if we better understand the physiology behind that. But basically what we found out using these models is that there's a lot of really neat things going on within the mammary gland. There's a group, he's now at the University of Missouri, that suggested there's some lactational programming that can occur that will carry over into the next lactation. So if you do something in such a way from a management perspective, in their first lactation, that might carry over into the next one that will make them a better lactator next time, or better able to get through a certain situation. We know that there are epigenetic mechanisms, and this is a really hot topic in the basic sciences right now, epigenetics, and what are we doing to our babies when they're growing, or what is the milk looking like as a result of something the mom ate while she was lactating? There's a lot of stuff going on here, and some of what Sam will talk about in her talk, we've seen some things with that, and we're trying to manipulate that using things like high-dose folic acid, and I'll let her discuss that in more detail. But there's a lot of good things going on that we're trying to understand to help from a dietary perspective during that. In the dairy world, there's this pre-calving time period where getting exactly the right kind of nutrients right in the last 3 weeks before they give birth really helps them from a maternal perspective, making milk, as well as having less stress when they give birth, and be better able to make milk without having any issues. And so we're really trying to kind of understand that better. So, as I said, cell number is really important. We have to have adequate mammary epithelial cell proliferation, and this is something Dr. English called me, I think maybe a year ago, and talked about a case she had with something that had happened during pregnancy, and there was low milk supply, and she had some issues with her hormone status during her pregnancy. And this is true. The endocrine part is just as important. So if something happens during the pregnancy that's going to prevent adequate cell proliferation, you simply just cannot make as much milk if you don't have the machinery to do it. And with that, the balance of the cell death. So keeping this tissue adequately growing as it should and really being optimized during the pregnancy is just as important for her to have a successful lactation. So the other thing that goes along with this is a lot of these extracellular matrix proteins. So one thing we think about, we're always saying more cells are better, and that is really good. But these cells have to interact with a lot of proteins that they're bound to, which allow them to change their shape and become milk-making cells. And so these extracellular matrix proteins are also really important in talking to those cells to say, let's make more milk. We can become the cell, this big, huge cell that has lots of fat organelles and mitochondria and Golgi to make all this protein and get it out and secreted. So it's not just having the cells, but it's also having this interaction with the extracellular matrix that's important. So what are these regulators of milk synthesis? So in 1961, like I said, sometime in the 50s to 60s, we knew that milk removal was really critical for the stimulation of milk, and this was somewhat locally regulated, and this was done in sheep and goats. Then they showed that if you had this removal of some factor, and I mentioned early on they thought it was a fatty acid, then it became Phil, which we'll focus on. When you decrease the amount of removal from these animals, secretion decreased overall. And then the group that kind of started doing this was in Scotland, and Linzell and Peaker were the two first kind of mammary physiologists that sort of started looking into this in more depth after this original paper, and they realized it wasn't oxytocin. So oxytocin wasn't driving this. And then they went on to do a bunch of different studies in a bunch of different species, and we'll get to when they figured out Phil and how this was regulating milk synthesis. So what they did show here was that frequency was not dependent on stretch, but they do have impacts on milk secretion, but there was this chemical present, and this was in the goat. And they did, like I said, they used a bellows, and then they also did isoosmotic infusions, so they would secrete a bunch of milk up into the teat because you can give an intramammary administration into these ruminant species because they have very large openings at their teat base, and so they'd put milk and then they'd put something that was similar in terms of isoosmolality, and it was usually like a sucrose, and it was just the milk that was causing the depression, not the sucrose. And so that was kind of how they got at the chemical part of it. And then what they started doing was they'd take the milk and they'd break it all up into different fractions using different chemical processes. And what they found was that lactose and casein were decreased when they took the whey protein part out. So they had their casein protein and then they had their whey fraction, and they would take cells from different animals. In this case, they were doing this in a rabbit, and they would put the whey protein either straight into the teat canal to go up into the tissue, or they'd put it on slices of the mammary tissue, and they would see decreased secretion of these various components of milk. Then they did this intramammary in goats, and they saw the decreased milk yield, and this was work largely done by Colin Wilde, who was a part of this group in Scotland. And then what they did find was that this had to do with suppression of protein synthesis, not fat. So it was some kind of protein effect, and they figured it had to be a protein that was present. And so then in 1995, they first described it as a small glycoprotein. And it is 22 to 23 kilodaltons in size. And that was all the information they gave. And at this point, we did know how to sequence proteins, like down to the amino acid. And they weren't providing a sequence. And so then they did this in a whole host of species. They did it in the Tamar wallaby, they did it in goats, they did it in sheep, they did it in cows, and they kept seeing this suppression in protein synthesis. And finally, pretty much most of the mammary gland biologists went to them and said, this is great, we understand this concept, and it seems to make sense. What is the chemical composition of this protein? And there was nothing. And when that happened, as I said, I think it was in the late 90s, they basically said, you either give us the identification or you're going to have to stop publishing. So if you ever go into PubMed and search Phil, you'll see that the last papers were in the late 90s of when they were published. And it was because they never produced the sequence. And I only know this story because my PhD advisor told it to me. But he always gives me these great tidbits of information because he's been involved in so many things. But basically, they just never provided it. And so people stopped publishing with Phil. But none of that really got translated out too much into the field unless you were in the scientific community that kind of heard about it. So we do know that this is a true situation. There is feedback inhibition of lactation. It's just that this protein, whatever it was, is probably a lot of different proteins. And since then, we'll go on to talk about a few of them. There's been several different proteins that have been identified that can have these milk suppressing effects at high concentrations on the tissue or in the milk. So while Phil itself was not something that we really understood because we don't really know what it is based on what that group was saying, it provided a lot of the groundwork for understanding that the mammary gland functions kind of on its own in lactation and is driving milk synthesis and secretion. So while there was some question as to what they were doing, and they kept seeing that it worked exactly the same across all species, which could be a little bit fishy. I mean, we're all mammals, but there are some nuances that are different. It did let us know that there was something going on in the chemical part of the milk causing these effects, and led us to a lot of really interesting research that has found several things that have provided a lot of knowledge and shown that you can really drive milk production once it's established without kind of messing with the hormonal status of an animal. So several of the things that we've found in the last 20 or so years is transforming growth factor beta, parathyroid hormone-related proteins, serotonin, lactoferrin, fatty acids, alpha-lactalbumin itself, one of the main milk proteins to make lactose. If you have lots and lots of it present in the milk, it will shut down milk synthesis. So a lot of things in high concentrations can have a negative effect. So basically it comes down to the more often you remove the milk, the lower the concentration, the less feedback. Pretty much with all of them. They all kind of work in that way. So as I said, this is what they had described Phil to be, a small acidic glycoprotein, somewhere in this range of size in the whey fraction of milk. And they did this in a lot of species, and we never really got this exact protein sequence, so they kind of stopped doing research on Phil itself. But a lot of people have gone on to show that our group, my PhD advisors group, my postdoc mentors group, and many others, that there are many things that are present that can have this effect. So there was some increased milking frequency studies, like I said, done in cows looking to optimize growth and increasing milk yield in the early lactation. What they saw were there was no changes in IGF-1, prolactin, growth hormone in cows that you have more milking frequency with. So just because you're emptying more or nursing more or milking more often, you're not changing the hormone status of the animal. And so this, again, corroborates what we've talked about. In 2004, my postdoc mentor discovered that serotonin was actually made by the mammary gland. And what they first saw was that in these high concentrations, so in low frequency events, you would have a decrease in milk protein expression, as well as have negative effects on the tight junction formation. But serotonin itself actually increases during lactation. And so far, we've been able to show that that is actually serotonin coming from the mammary gland and not from the gut. So it is a significant source during lactation. But like all things, concentration dependent. And in another group in 2000 looked at de novo fatty acids. So they're in ruminant animals because of the big bacterial fermentation vat they have at the front of their stomach. They can produce these things called trans fatty acids, which I'm sure everyone's like, oh no, the trans fatty acids. These are not the fake ones. These are the natural ones that occur because they're breaking down grass in their rumen. And sometimes, depending on how you feed the animal, you can get overproduction of these fats that will depress milk synthesis. And so that was one thing that they found in the ruminant species, that there are particular fatty acids that can also have a negative effect on milk production. We showed in my PhD that if you gave high concentrations of serotonin, really, really high, that are super physiological, we would see a reduction in milk production. In 1992, it was determined that parathyroid hormone-related protein was detected only in mothers who breastfed, and this is a specific hormone that regulates calcium metabolism during lactation only, and also in metastatic breast cancers or prostate cancers that go to the bone. And humans will secrete more milk when they breastfeed more often, so this was tied together, and they didn't see this hormone in mothers that were bottle feeding because they weren't making milk. Nonetheless, this particular portion of research showed that this PTHRP was made in lactating mothers only and not the bottle-fed mothers, and we're still pretty puzzled as to what PTHRP does for the baby, but it's in the milk at like 10 times or 100 times higher than it is in the blood. But we do know it coordinates calcium metabolism in the mom. And then they went on to see there was a lot more bioactive factors present in the milk. TGF-beta at high concentrations would suppress protein expression. They showed that growth hormone didn't act locally, but it was rather through the IGF production. So if you have growth hormone, you have more IGF-1, and you get more mammary epithelial cell proliferation. And I'll talk more about IGF-1 and insulin in the next talk. And then in 2012, we discovered that serotonin was modulating calcium homeostasis through PTHRP production. And that's a lot of what Sam will be talking to you about. So what are ways we can increase milk secretion? Milking frequency, obviously, is a go-to. More often, you should get more milk production. We know that at least in ruminant species, somatostatin or IGF-1, growth hormone is really what's driving a lot of this milk production, and it's because growth hormone is really good at coordinating metabolism. So it's the growth hormone saying we need more fat, or we need more glucose, or we need more protein. It's just coordinating the mom's metabolism so she can drive her nutrients better to the tissue to make more milk. And that's really what growth hormone does for lactation. Cell proliferation is really important, so you have to have adequate cells present to make more milk. And differentiation of the cells is also important, so this interaction with that extracellular matrix. Serotonin and parathyroid hormone-related protein appear to have quite a bit of influence, and we keep finding more and more things that serotonin does. So hopefully it'll keep me going for the rest of my career, hopefully. We know prolactin's really important. It's the critical galactagogue in most species, but in ruminants as well, it's critical for setting up lactogenesis. So you do have to have prolactin. It just doesn't maintain the milk production like it does in non-ruminants. And then your balance of apoptosis, doing things to the tissue that are going to cause more cells to die than be alive, clearly you're going to have less milk production. And then casein, there was a casein phosphopeptide, which was another one of these local factors that can have negative effects depending on concentration. So as I said, we've been spending a lot of our time looking at serotonin in the mammary gland. This has been since the beginning of my career as a grad student. We've showed that it affects tight junction status in the mammary gland, so we know at high concentrations it'll cause the tight junctions to go down, be disrupted. And we've seen this in human cells, mouse cells. We saw this in a cohort of women that were on really high doses of SSRIs. And we also know that it affects calcium homeostasis. But the thing that we also know is there's like 4 or 5 other receptors in the tissue that we have no idea what serotonin's doing to the mammary gland through those receptors. So we're still trying to kind of figure all this out. And some of it is also related to some obesity research we're doing, but I won't talk about today. So kind of our hypothesis with serotonin and calcium is that it's driving calcium uptake by the mammary gland through these channels that suck calcium into the cell. We've seen that it increases calcium concentrations into the milk. So when we give a precursor to serotonin, it'll cause more of it to go into the milk as well. And the thing about coordinating calcium metabolism in the mother is it's a negative feedback response, like a lot of things. So you put more calcium into milk and she's able to pull more calcium from the bone. And we know that that's the major source of her calcium for keeping herself well as making milk during lactation. So this drives this negative feedback, and we get more PTHRP being made by the mammary gland, and then PTHRP can drive this calcium release from the bone tissue and restore calcium homeostasis. So this is a lot of what Sam will be talking to you about, and as I said, the major focus of our lab. But in summary, basically we have all these different factors that affect milk production. We know serotonin's interacting through its receptors. It triggers calcium-related events as well as tight junction status. And those are all really important to regulate maternal metabolism as well as milk secretion and synthesis. And so that's kind of the main thing we've been focusing on. We know prolactin's really important, and it can work through this protein synthesis pathway, which I'll talk some about in my next talk with insulin and IGF-1 and what's really important in the mammary gland. And it's also really critical for stimulating this other protein complex that makes milk protein specifically, the JAK-STAT pathway. TGF-beta and alpha-lactalbumin, they can have a negative effect on milk protein synthesis. So if you have milk building up, their concentration increases, and that can have negative feedback that will suppress milk protein production and lead to apoptosis. And then we have growth hormone through the IGF-1 pathway that is going to be important in proliferation of the cells as well as coordination of the mom's metabolism to have efficient milk production. And so with that, I'd like to thank you guys and answer any questions you may have. And this is us.
So Allison and Kathy will move around and bring up some questions. We have a few minutes for questions. But since I'm here and I have my questions, I can ask those. So how many of these factors do you think are diet related for mother? Because, for example, I have patients who swear to me that when they eat red meat, their supply goes down. And if they eat like lean meat, it's better. So then I was wondering, well, maybe it's the animal fat, because I hear that also when they go to like Culver's or somewhere else where they have like custard, which is, for those of you who are not from Wisconsin, custard is all, a huge thing here. I see women will say that they notice more plugs and that sometimes their supply goes down.
That's an interesting question. I don't know that anyone's specifically looked at that. We do know at least, so potentially these could be obese moms maybe, or is this just in general? Because for sure, on the obese side, we know they struggle at the beginning, and even in our rodent studies, they're okay once they get past that beginning phase and they actually make more milk than the ones that are lean often, at least in our controlled experiments. But I don't know if it's dietary related. We do know, at least in a ruminant, which is very different because they have that big fermentation vat, and when you feed them different things, it can cause the production of what is trans 10 cis 12 CLA, and that's one that can cause milk fat depression in dairy cows, which decreases milk yield. So potentially there could be things like that happen. If you affect the way they digest something, they might produce something else that then causes a negative feedback on the tissue. And we know that in cows, but I don't know that anyone studied it in humans specifically.
Okay, great. Okay, so do you want to read that?
Yep. Are humans capable of asynchronous concurrent lactation? That's a great question. I don't know that anyone's taken a milk sample and compared, but I have seen reports where there's a mom feeding a 2 or 3 year old on one side and then also feeding the baby on the other side. So my guess is probably. I don't know that anyone's though taken samples, and it would be really cool to do so. I would love to do that and actually look at the differences between the two, but it really is, there is some kind of signal that is communicating with the baby and with the pregnancy that might cause colostrum production in one or both, but that'd be really interesting to study.
So there's a couple questions about regeneration, like the difference in milk, if there's no resting period.
Okay.
Yeah.
So what happens if breast tissue doesn't have rest and regeneration between lactations? This has been really well studied in dairy cows, actually, because we have an optimal dry period length. And we like to give them 45 days at least resting with no lactation. And what they've shown is that if they stay continuously lactating through their pregnancy, they will see anywhere from a 10 to 30% reduction in their next cumulative milk yield because the tissue hasn't been able to recover. You haven't given the cells enough time to die off and replace. So you just have old cells basically working that eventually are going to die, and then you're having to lactate at the same time, and so they don't quite get the repair time that they need.
Interesting. Okay. So let's see.
What is the difference between a mom's mammary gland from a mom with a 24-week NICU baby and a term baby? That's a great question. So the primary time period in which you get the most proliferation and differentiation of that tissue is the third trimester. That's when things really start to go like gangbusters. So if you're having a baby at 24 weeks versus full term, you're missing a big part of that proliferation, and probably stage 1 lactogenesis, because term moms or term animals will already have some milk present in their tissue. And if you have a baby too early, you may not be to that stage yet. And so it's going to take some time to kind of get that process going. And you may not have as many cells either to make as much milk as you would if you'd gone the whole pregnancy. Is there anything that can be done during pregnancy to set up the breast for optimal milk production, i.e. if women had low milk supply with the previous pregnancy or no breast growth during pregnancy? I'm not really sure about that one. I would guess there would be some kind of hormonal treatment. However, I'm not sure that a woman would want that or not. That's the thing that you always run into with some of these. What can you do to fix it? It might mean you might need more estrogen or progesterone or something during your pregnancy to maybe help better differentiate the tissue.
Like women who have insulin resistance and they're given metformin during pregnancy?
Yes, so that's a good one because actually I mentioned it in the next talk too, is women that have high insulin resistance and metformin. And some of Lori Nomsen-Rivers' work from Cincinnati, they're showing that potentially this could help because the question is it insulin or IGF-1? Probably both because they can work through different receptors at different concentrations. And so if you have the inability to respond to insulin, you may not be able to have adequate lactogenesis, but the mammary gland also, glucose uptake by the mammary gland is not insulin dependent. But there might be some differentiation problems that could occur as a result of that as well. And so if you took metformin, she's showing that, I don't know if she's published it yet, I talked to her a couple weeks ago at EB, but she's seeing that an improvement in milk production with the metformin.